In this lesson on meiosis, we're going to follow the chromosome number. All right, so chromosomes, those strands of DNA and protein found inside the cell's nucleus, are the carriers of genes. And these genes are located in very specific positions on those chromosomes. So almost every cell in our bodies, and your body as well, has two copies of each chromosome, one you got from your father and one you got from your mother. Now, these chromosomes, they have the same genes, and that's what we call homologous. Homo means same, and log, like a homolog, is something that's the same. So humans have actually 23 pairs of chromosomes, or 46 total. Now, the example we see here on screen is actually from a fruit fly, and the fruit fly's body cells are what we call diploid, and the gametes are called haploid. Okay, so the somatic body cells are diploid and the gametes are haploid. Now, we can also designate them by a code, if you will. So the reason we use the number N is because it's the number of chromosomes for that particular organism. Now, fruit flies don't have 23 chromosomes. As you can see here, they actually only have four chromosomes. In the haploid cell, you see one chromosome here, the red one. These are nicely color-coded to make it easy to see. The green one, that's 2. The orange one, that's 3. And then the little purple one is another chromosome that's all part of the fruit fly's DNA. So their diploid cells would have 2 of each of these, which would give it a total of 8. So the fruit fly, its chromosome number is 4. So a diploid cell that's 2N would be 2 times 4, which would give us 8. Whereas the haploid cell only has one copy of each gene, the 4 genes, so its number is just 1N or N. Okay, 1 times 4 is 4. So let me ask you, are most of the cells in our body haploid or diploid? What do you think? Well, if the only cells in our body that are haploid are the gametes, one would have to surmise, I would hope, that we have more diploid cells than we have haploid cells, because only the gametes are haploid. But all the rest of our body cells are diploid. Now, before mitosis occurs, is the parent cell haploid or diploid? What about at the end of mitosis? Are the daughter cells haploid or diploid? Well, the answer to that is both are considered diploid. Now, the tricky thing is at the end of mitosis, there should actually be double the amount of the doubled amount, right? There would be 92 chromosomes total, 4 pairs. But then they would get split up into the daughter cells and they would be diploid. So diploid really refers to any cell that has at least one extra copy or one set of pairs of the chromosomes. Okay. The only ones we call haploid are the ones that have only one set of the chromosomes. Okay, so as we continue looking at this particular image, what you see is these 2 red ones here. Because they're identical, they're referred to as homologs. Okay, these are the homologs. Okay, these 2 green ones would be homologs of each other. They would not be homologs of the red ones. They are different. They contain different genes, different Okay, even these two little purple ones would be a homologous pair or homologs. Okay, so why do you think this particular cell has replicated its DNA? Well, if you're thinking, must have something to do with the cell cycle, because we know that during the cell cycle, the DNA gets replicated, well, you'd be on the right track. The cell has just replicated its DNA so it can enter the process of cell division. Okay, let's take a quick review of mitosis, because as we move forward and get into meiosis in the next lesson, we're going to see that meiosis is pretty similar to mitosis. So it would be good to just kind of brush through this real quickly again. The first stage of mitosis is prophase, and that is when the nuclear envelope begins to break down and the spindle fibers begin to form from the centrioles. And they're going to connect to the centrosomes of the homologous pairs. And there's that word again, homologous pairs. Then metaphase is where the chromosomes are lined up across the middle of the cell, preparing for separation. In anaphase, those homologous pairs are going to be pulled apart in anaphase. And then in telophase, we get the nuclear envelope beginning to reform and the cytoplasm just starting to pinch in a little bit. And then we hit cytokinesis in it, and it completely splits into two separate cells. So let's think about the significance of a cell being haploid or diploid. I'd like you to consider what kind of cells would need to be haploid and why. It's not like we haven't talked about this. You should conclude that the gametes, the eggs and the sperm, need to be haploid so that when fertilization occurs, the offspring don't end up with two full sets of chromosomes from each parent, meaning four sets of chromosomes. So the process that produces these sex cells is called meiosis. It's spelled weird, but we say it meiosis. So this process that produces the sex cells, therefore, needs to divide the nuclei and shuffle chromosomes so that the resulting cells have half the normal chromosome number. Now, before meiosis begins, every chromosome is copied. So the cell has four copies of each chromosome. And you can see those structures here. Okay, and now just like regular mitosis, in prophase 1, the nuclear envelope has begun breaking down, the spindle fibers are being formed, so forth and so on. And then in metaphase 1, the pairs, remember there's four pairs, they get lined up in the middle. And then in anaphase 1, they get pulled apart. And then we have telophase 1 and of course cytokinesis, where the cell actually split into two. Now those two new daughter cells are now going to move into what's called meiosis II. So remember that at this point, each of those two cells has 46 chromosomes. If we're talking about humans, if we're talking about fruit flies, it would be 8. Okay, the 2n number. So in a sense, they're not gametes yet. They're still diploid. They need to become haploid. The way that happens is that without going into G1 phase or S phase, where the DNA gets replicated once again, they skip that and they go right back into prophase, the first phase of mitosis. We call it prophase 2, though, because we're working within the realm of meiosis, where we're actually producing gametes as the end result. You see those down here. So prophase 1, prophase 2, just like prophase 1, has the nuclear envelopes breaking down, the spindle fibers forming, starting to attach to the centrosomes of the homologous pairs. And then in metaphase 2, they get lined up in the middle. And then in anaphase 2, they get pulled apart. So those steps are very, very similar, if not nearly identical, to mitosis. The difference is there's only one set of chromosomes, one set of chromosomes, which means one pair, right? As opposed to four pairs. So that one pair then gets pulled apart in anaphase, and in telophase, Remember, we started with 2 at the beginning of meiosis II, 2 cells. By the end of telophase 2 and cytokinesis, those 2 cells are each going to split in 2, resulting in now 4 daughter cells, and they are only going to have 1 copy of the chromosomes. So now they are haploid daughter cells. So as you look a little more closely at each of these stages, I want you to think about how these stages are different to and similar to the stages of mitosis, kind of as I've just described. All right, so here we see a cell that's in prophase 1. So this is meiosis. We know that because it says prophase 1. That tells us we're in meiosis. And this is kind of a close-up here as we kind of draw out these particular homologous pairs. And we're going to see that during prophase 1, there is something that occurs that is not similar to what happens in mitosis. So we start out with some familiar structures. First, we have the centrioles, okay? They're going to send out the spindle fibers. So we see those forming here as well. And the nuclear envelope has broken down. Here are our chromosomes. And we also have the centromeres, which is where the spindle fibers attach to pull things apart. So in prophase 1 of meiosis, each replicated chromosome pairs with its corresponding homologous chromosome. This pairing forms a structure called a tetrad, which contains 4 chromatids. Okay, and here is one of the tetrads. Now, as the homologous chromosomes form their tetrads, they undergo a process called crossing over. First, the chromatids of the homologous chromosomes cross over one another. They kind of just overlap. And then the crossed sections of the chromatids, which contain alleles, are actually exchanged. Crossing over, therefore, produces new combinations of alleles in the cell. So how does crossing over affect the alleles of a chromosome? Well, during crossing over, the alleles can be exchanged between chromatids of homologous chromosomes to produce new combinations of alleles. And during metaphase 1 of meiosis, the paired homologous chromosomes will line up across the center of the cell. Here we see a pair of homologous chromosomes. And we can also see that some of the colors are inconsistent. We have some yellow on the red one, and we have some red on the yellow one. So that's showing, illustrating essentially that some crossing over has occurred. So how is metaphase 1 of meiosis similar to metaphase of mitosis? Well, the chromosomes line up along the center of the cell in both forms of metaphase and metaphase 1. So what do you think the pink and yellow chromosomes and pieces of chromosomes represent in the diagram? Well, the yellow pieces were originally from one of the organism's parents, and the red pieces are originally from the other parent. So how many chromatids are in this cell? Are the sister chromatids identical? Well, there are 8 chromatids in this cell. And they are 1 here and 2, 3 and 4, 5, 6, 7, 8. Are the sister chromatids identical? Well, not any longer, because crossing over has occurred. And when that occurs, there's an exchange of alleles. So now they're different. All right, now during anaphase 1, the spindle fibers are going to pull each of the homologous pairs apart towards opposite ends of the cell. So here we see the sister chromatids. So what effect does anaphase 1 have on the chromosome number of the cells that we ultimately result? Well, it cuts the chromosome number in half. The next phase is telophase 1, and this is where a nuclear membrane forms around each cluster of chromosomes. And then cytokinesis follows telophase 1, forming 2 new cells. You see the nuclear envelope represented by the dashed line in the illustration. So, how does anaphase 1 and telophase 1 of meiosis differ from anaphase and telophase in mitosis? Well, the chromosomes are still doubled at the end of meiosis 1. The number of chromosomes needs to be halved. So, what is the end product of meiosis 1? Well, the end product of meiosis 1 would be the two new daughter cells. So, are these daughter cells haploid or diploid? Well, each of those two new daughter cells would be diploid. All right, now we get into meiosis II. And so all of the stages of meiosis II are going to have that 2 designation at the end. So prophase 2, metaphase 2, anaphase 2, telophase 2. Cytokinesis doesn't have the designation of 2. It's just cytokinesis. It's always the same. All right, so the two cells at the top of this illustration here, take a look at those. These cells have two one end chromosomes, but there are 4 chromatids. So, unlike the first division, neither of the cells has gone through a round of chromosome replication before entering meiosis II. Okay, it skips the S phase. During metaphase of meiosis II, so metaphase 2, the chromosomes line up in the center of each cell, just like in metaphase 1 and metaphase of mitosis. As the cell enters anaphase, though, the paired chromatids begin to separate. And that too is like anaphase 1 as well as mitosis anaphase. But the final 4 phases of mitosis, they just really are very much like meiosis 1. However, the end result, because there's 2 cells going through that process, the end result ends up being 4 haploid daughter cells. So, how does anaphase 2 differ from anaphase of mitosis, and how are they similar? Well, in anaphase of mitosis and in anaphase 2, the sister chromatids do separate. But in anaphase 2, there are no homologous chromosomes left. So the cell is haploid, not diploid. So, what is the chromosome number of each cell? The answer is 2. What is the chromosome number of the original parent cell? Well, according to this illustration, it would be 4. So, how many haploid or one end daughter cells are produced at the end of meiosis II? Well, hopefully you said 4. So, meiosis results in cells that have half the normal chromosome number. So, what are some differences between meiosis I and meiosis II? Well, the simple answer would be that homologous chromosomes separate during meiosis I, but not during meiosis II. The centromeres and the sister chromatids separate during meiosis II. So, with this illustration, we can kind of compare and contrast mitosis and meiosis. And I'd like to draw particular attention to the phases in meiosis where genetic recombination occurs. For example, in prophase and mitosis, the replicated chromosomes don't pair up, whereas in prophase 1 in meiosis, the replicated chromosomes do pair up. They pair up with their homologous chromosomes, the homologs. And once that happens, the process of crossing over takes place. So, as we walk through the diagram, take a note of the differences in the lining up of chromosomes, also the number of chromosomes each cell contains, and how the chromosomes separate into new cells. So, in this particular cell, in mitosis, we have 1, 2, 3, 4, 5, 6, 7, 8 chromosomes. Okay, that's the 2n number. They're going to line up in the middle and then get separated. So now we have 1, 2, 3, 4, 5, 6, 7, 8. Okay, and then they get separated, and cytokinesis pulls them apart into 2 new daughter cells that each has 4 chromosomes. Now, in mitosis, when the 2 sets of genetic material separate, each of the daughter cells receives 1 complete set of chromosomes. But in meiosis, the homologous chromosomes line up and then move to separate daughter cells. Mitosis does not normally change the chromosome number of the original cell. This is not the case, though, for meiosis, which reduces the chromosome number by half. Mitosis results in the production of two genetically identical diploid cells, whereas meiosis produces four genetically different haploid cells. So we can conclude from this that ultimately mitosis and meiosis both ensure that cells inherit genetic information. And also, both processes begin after interphase once chromosome replication had occurred. However, the two processes differ in the way that the chromosomes are separated, as well as the number of cells produced and the number of chromosomes each cell contains. So in mitosis, the number of chromosomes contained in each cell is the 2n number, which in this case is 4. Whereas in meiosis, it is just 2 chromosomes. That is the chromosome number in this particular, I think we're still looking at the fruit fly, okay, in the fruit fly cells. All right, now let's think back to Mendel's principle of independent assortment. You should recall that Mendel found that genes that are located on different chromosomes assort independently from other genes. But what about genes that are located on the same chromosome? Well, researcher Thomas Hunt Morgan back in 1910, so really only about 110 years ago, he considered the question of whether genes on the same chromosome are generally inherited together. So he studied the genes of fruit flies. Of course, we had the technology available to do that at that point. And what he found was that many genes were linked in a way that seemed to violate independent assortment. So here in the diagram, we can see that during crossover, there are chunks of chromosomes, not individual genes, that are actually exchanged between the chromosomes. So this means that some genes are likely to travel together. That is, they are linked. This is what Morgan found in his fruit fly experiments, that chunks of genes sorted independently, but genes within certain linkage groups were inherited together. One of Morgan's students, his name was Alfred Sturtevant, he wondered if the frequency of crossing over between genes during meiosis might be a clue to the gene's locations. So Sturtevant reasoned that the farther apart the two genes were on a chromosome, the more likely it would be that crossing over would occur between them. Now, do you think crossovers are more common between genes that are far apart or genes that are close together on a chromosome? Well, crossovers are actually more common between genes that are farther apart. So Sturtevant reasoned that he could use the frequency of crossing over between genes to determine their distances from each other. And using this logic, he was able to create a diagram like the one you see here, and it is called a gene map. And it shows the location of a variety of genes on a chromosome. In this case, it's chromosome 2 from the fruit fly. The genes are named after the problems that abnormal alleles cause, not after the normal structures. So, where on the chromosome is the purple eye gene located? Let's see. We look for it right here. Boom. Purple eye. We are going to go to its location, and the location is at 54.5. So which genes are likely to be inherited together and why? Well, if you look at the star eye, which is located at 1.3, and you look at dumpy wing, which is right next, which is actually at 13, they would likely be inherited together because these genes are so close together on the chromosome. So the chance of that crossing over would separate them is smaller.